Hello, hello students. In previous lecture, we studied growth, growth hormones or growth regulators. We studied growth hormones, auxins, physiological effects and applications of auxin. Now, today we are going to study another growth hormones. First one, gibberellins. It is another growth promoting hormone and is abundant in root tip and developing seeds. Gibberellins were first isolated from the fungus Gibberella fusiculae by Japanese scientist Kurosawa in 1926. The crystalline form of gibberellins were isolated by Yabuta and Sumuki in 1938 from the fungus culture. It is synthesized in young leaves, seeds, root and stem tips. These are synthesized from mevalonic acid. GA3 is the most common and biological active form. Now, physiological effects and application of gibberellins. Dormancy of bud can be broken by gibberellin treatment. It can promote seed germination in cereals like barley and wheat by synthesizing hydrolyzing enzyme amylase to produce sugar. It also promotes bolting, that is, elongation of internodes. It causes parthenocarpy in tomato, apple, and pear, and flowering in long day plants. It is used to increase the fruit size and bunch length of grapes. When gibberellins are applied on genetically dark plants like maize, the stem rapidly elongates and acquires the height of normal tall varieties of maize. Usually, it inhibits growth of root, delays sensations, and prevents abscissions. It also breaks dormancy of seed. Application of gibberellins causes production of male flowers on female plant. Next, growth hormone cytokinin. It is another growth hormone that promotes cell division. Latham coined the term cytokinin. The first cytokinin was discovered by Cook and Miller in 1954 during investigation of nutritional requirements of callus tissue culture of Nicotiana tabacum. First natural cytokinin was obtained from unripe maize grains by Latham. It is known as G10. G6-benzyl adenine is a synthetic cytokinin hormone. 7 different types of cytokinins are recorded from plants Natural cytokinins are also reported from plants like banana flowers, apple and tomato fruits, coconut milks, etc. Now next, physiological effects and applications of cytokinin. Beside cell division, it also promotes cell enlargement. A low ratio of cytokinin to auxin induces root development, but a high ratio causes buds and shoot to develop. It promotes the growth of lateral buds and controls apical dominance by cell division. It also breaks dormancy and promotes the germination of seeds. It induces RNA synthesis and formation of interfascicular cambium. In this way, we study physiological effects and application of cytokinin. Next, another growth regulator, ethylene. It is the only gaseous growth regulator. Crocker and Denny in 1924 reported ethylene is effective in fruit ripening. Gain in 1934 established that plants naturally synthesize ethylene. The most widely used compound as a source of ethylene is ethefone. It is synthesized in roots, shoot apical meristem, ripening fruits. Now next topic, physiological effects and applications of ethylene. Ethylene promotes ripening of fruits like bananas, apples and mangoes. It stimulates initiation of lateral roots in plants and breaks the dormancy of bud and seed. Ethylene inhibits the growth of lateral buds and causes apical dominance and retards flowering. It is associated with the enhancement of process of sensations of plants organs. It inhibits flowering in most of the plants except pineapple. It causes epinasty, means drooping of leaves and flowers. It increases activity of chlorophyllase enzyme, causing degreening effect in banana and citrus fruits. Degreening effect means it is a process of decomposition of green pigments in fruits, usually by applying ethylene. In this way, we studied physiological effects and application of ethylene. Today we studied growth hormone gibberellins, physiological effects and application of gibberellins, another growth hormone cytokinin, physiological effect and application of cytokinins, growth regulator, gaseous growth regulator ethylene, application and physiological effects of ethylene. In this way, these are different growth regulators. Now, next growth regulator, abscisic acid, we will study in next lecture. Thank you.